Well, first of all, I'd like to say thank you to the organizers for giving me this opportunity to visit such a beautiful city and to attend such a wonderful conference and to have the opportunity to share with you today some of the work that we've been involved with in our lab. There we go. So to begin with, this slide works equally well whether we're talking about obesity, diabetes, heart disease, any of these complex phenomena. But I've got it labeled obesity because I think one of the things that's really important when you're talking about trying to understand the genetics of any complex phenomena is how well we go about defining the phenotype or the phenomenon that we're interested in. And this old tale about the blind man and the elephant, I think, is very apt in that regard, because depending upon what aspects of that entity you look at, you get very different impressions. I mean, the whole idea being, is obesity the result of overintake or a failure of appetite regulation? Is it a failure, on the other hand, of energy expenditure? And now as we realize that the adipocytes themselves, or the adipose tissue, is such an active endocrine organ, the actual functioning of the adipose tissue in relation to the risk for a variety of cardiometabolic diseases becomes equally important. So when we talk about trying to understand the genetics of obesity or any of these complex diseases, I think we've set ourselves a very big task, and what we really need to do at times is go back and refocus on what types of phenotypes we're going to look at. With that being said, however, from the standpoint of a genetic epidemiologist, I think this is a pretty simple cartoon of what the challenge is. And that when we think about whatever phenotype we want to focus on, whether it's BMI or any of these quantitative measures of disease, that we can look around the room, and the thing we notice, of course, is there's a wide range of variation in this phenomenon. And as geneticists, of course, what we want to try to do is get from what we observe over to at this end of what the underlying genetic contribution is. And I believe that this maze here in the middle is an apt representation of that course, because it's not a straight line from one to the other, but it is rather circuitous, and there's a lot of dead ends and such at times. Well, one of the things I want to talk about today are some of the results that are currently out there, and what I won't do is spend very much time talking about the results from GWAS, because I know Dr. Luce will do an excellent job of presenting that information later this afternoon. But I do want to bring out a couple of points that I think are worth revisiting. And that basically what's happened over the last decade to a decade and a half has been a movement toward genome-wide approaches to trying to understand the genetic contribution to complex phenomenon. And we really began this effort by doing genome-wide linkage studies. We've now progressed to genome-wide association studies as well. And science being as it is at times with fads, everybody drops one and runs to the next, and in the end what we find out is, well, they all have their benefits, they all have their risk. But there's a couple of things I think are worth pointing out, because I want to focus on the linkage results actually today. And one of the things I think it's worth doing is sort of contrasting these two phenomena. On the one hand, we have to be upfront and be intellectual about is the fact that there's a major difference between the concept of linkage and association. And that is, linkage is inherently a genetic phenomenon. Association is a statistical phenomenon. While association gives you an indication of perhaps underlying genetic effects, it in and of itself is not genetic in origin. When we talk about linkage studies, these are family-based studies. And this is important because, again, if you go back to the principles of Mendel, it's the concept of transmission of information from generation to generation in a predictable pattern. Both of these methods, or both of these types of approaches, of course, allow us to utilize quantitative and as well as qualitative phenotypes, although I tend to lean toward the end of the quantitative feeling much more informative. And again, in terms of the type of genetic markers that are used in linkage, of course, originally started out with the STRs. Of course, now we've moved to SNPs as well. And the information, though, that we're exploiting in our attempt to try to identify these contributions comes from segregation information. Again, like I said, that transmission of genes from one generation to the next. And this is what's referred to in this case as identity by descent. And this is the concept that I and one of my relatives share a particular allele of a gene because we inherit it from the same ancestor. That it's identical because it's come down through descent lines those way. And we can contrast that with association in that those are typically case control studies, although as you see, and increasingly these can also be done in family studies as well. But the big difference is in the information content that's being used. So a linkage study, as I said, is based on the identity by descent information, and in association, it's identity by state, and that is that I and any one of you may share the same version of a gene because we share it because it's at the same state with one another. Now, the secret that's often not expressed behind all of this, of course, is that the reason association works and is using identity by state is that if we go far enough back in any population, identity by state and identity by descent become the same. When we do it with linkage, we just have a much more direct measure of it. So with that as the setup, sort of look, what have we found? So hopefully we've done a little better than Diogenes in terms of finding an honest man, and I think we have. So, like I said, I won't spend much time with the GWAS information. As I said, Dr. Luce will talk about that, but there have been many genes identified, several with very strong replicated findings, and many more continue to come out. Here, just showing the FTO gene and the melanocortin-4 gene, which have come up repeatedly in multiple studies. I have the melanocortin-4 gene highlighted in red, so I want to come back to this because this is not only detected in association studies, but it also has been identified through linkage studies as well. So, using or quoting from one of my favorite American authors, Mark Twain, when it comes to talking about linkage, I think the reports of my death have been greatly exaggerated. And what you're beginning to see is that as we realize is these GWASs are completing and coming along, that we're still not finding everything, and there is sort of a resurgence of going back and using the family information and the family data at the same time as well. So what I want to do with the remaining time this afternoon is give you bring up some highlights from some of the studies that I'm directly involved in, and of course, again, being a Mendelian-type geneticist, I've arranged these in the terms of a pedigree somewhat. And the diet and genotype in primate atherosclerosis, this was actually the first study begun at our institution over 30 years ago to focus on the genetics of common complex diseases, specifically heart disease. And it's really sort of the progenitor study in terms of its format for all the other studies we've developed. And so now, which was directly begun out of that, was the San Antonio Family Heart Study, which is now entering its 12th or 13th year. And again, the focus here being these are the recruitment of large extended families. Also now involved with people at Wake Forest University with their vervet colony, so we're continuing to do work in primates as well as the humans. But a number of other studies really are the direct outgrowth of the San Antonio Family Heart Study in terms of format. Our Viva La Familia study, this is in collaboration with Dr. Nancy Butte at the Baylor College of Medicine. This is focused on identifying the genetic contribution to risk for diabetes and obesity, metabolic complications in Hispanic children living in Houston, Texas. The Gokadan study is one that I direct on the genetics of coronary artery disease in Alaskan natives. I'll talk more about that one in a little bit. And then we also have a couple of studies in Native American populations, the Zuni Indian study with the Zuni population in New Mexico, as well as the Strong Heart Family Study, which is multiple Native American populations from the central part of the U.S., mainly Sioux and Dakota. We also have a collaboration with colleagues at the Sultan Qaboos University in Oman with a large population of Arab Bedouins. So to begin with, let me just give you highlights from the San Antonio Family Heart Study, and just one in particular. This is actually sort of an old result at this stage. This is one we've published about 13 years ago now, but this was really the first big finding from a genome-wide linkage study for obesity-related phenotypes in humans, and this is for serum leptin levels, circulating leptin levels in the San Antonio Family Heart Study. And one of the things to point out here when we talk about linkage results is that the y-axis here is the LOD score. That's a log odds ratio, so that's an order of magnitude for each unit of increase. And the typical dogma is you need a LOD score of 3 to conclude significant linkage, and what you can see, of course, here is we have a LOD score of well over 7. So tremendously strong evidence for a gene in this region contributing to the variation we observe in circulating leptin levels, as well as some associated fat mass measures in this population. But of particular interest is lying within the 1 LOD unit support interval of that linkage signal in this area here is the gene POMC, or proopiomelanocortin. And so it was one of the ones we really first began to follow up and work on, and this is where the work really over the last year or two has been focused in this study. And that thus far we've gone in, we've resequenced the POMC gene in a number of the individuals in the population. We've identified a number of common SNPs within that gene. We've typed those now in all the participants in the study. And one of the things in doing that, of course, is we've identified now through Bayesian quantitative trait nucleotide analysis at least 4 SNPs that seem to have at least putative functionality in terms of driving this linkage signal that we detect. Now, interestingly enough, that when we actually go back and redo the linkage analysis conditional on those 4 SNPs, we significantly decrease that LOD score. We drop it down to just a little bit over a 1, or a 2 in this case. And what that means, of course, is that the SNPs we've identified go a long ways to contributing or accounting for the variation that we're observing. But the fact that it doesn't go to 0 also tells us there's still probably some additional variation to be looked at out there. So what we're doing now, of course, is we're expanding our sequencing, going further out on both sides of the POMC gene, but we're also expanding it to look at there's actually a couple of other potential genes that may be involved, so we're beginning to sequence those too in that same region. So the Viva La Familia study, though, is one that I want to spend a little time on. And this one, I think, is a good example, going back to the comments about the blind man and the elephant, that one of the things that's been a real strength of this study is that the Baylor College of Medicine, the pediatric center there, it's a USDA-funded facility, has a phenomenal capability for phenotyping. So not only do we have the typical measurements of body composition and circulating proteins and such, but we have very good data on 24-hour whole room calorimetry on all these children who are participating. So there's about 1,300 kids in this study. And so while we have many signals here that are quite interesting and we're currently pursuing, I want to focus this afternoon a little bit more on our melanocortin-4 signal. So one of the first signals that popped up in this study was melanocortin-4, and this is when we looked at this wide range of markers of energy expenditure from the calorimeter. And one of the first questions, of course, that I asked when we saw that result was that we know that there are a variety of single or rare mutations in the melanocortin-4 gene that account for a large proportion of the early onset sort of massive childhood obesity that's out there. And what I was hoping was we hadn't just found another one of those. So one of the things we did was we removed those, we looked at the data, and what we found, indeed, there was one family, one set of brothers that were having an undue influence on the rest of the analysis. They were considerably much more obese than all the other participants. And we actually took their DNA, sent it to colleagues in Cambridge, Sadaf Faruqi and Steve O'Reilly, to let them look at that, since that was sort of a little bit more along their interest. And while doing that, went back and redid our analysis. And what we found was that while removing that one set of brothers did decrease our initial LOD score, it still remained significantly over 3. At the same time, as Sadaf and Steve were looking at the set of brothers that we sent to them, they were able to identify indeed a polymorphism in the melanocortin-4 gene of that particular set of brothers that was very similar to what had been seen for the other known variations causing early and severe obesity in children. But what we've done, like I said, is we repeated the analysis with those boys out of it. We still get a significant linkage signal, and we've now gone in and resequenced the melanocortin-4 gene in our remaining sample. And what we've come of that is we've now identified 2 SNPs, 1704 and 2132, both of which have strong statistical support for putative functionality. 1704 influencing sedentary activity, moderate activity measures, while 2132 impacting total energy expenditure and sleeping metabolic rate. So what we're after, or the process we're in now, of course, is going back and doing even more extensive phenotype or sequencing in these genes, make sure we've got all of them, as well as beginning some of the functional assays. I think what's important about this is I think this is an interesting example of the fact that the melanocortin-4 gene is known to produce now both rare and extreme forms of obesity, as well as having variants that contribute to much more normal, everyday varieties of variation in body weight. And then I want to finish up here, talk briefly about our work in the Alaskan Eskimos. And I think this is a particularly interesting population for a number of reasons. This study is focused in the Norton Sound region of Alaska. I don't know if you're familiar with Alaska, but this is Nome, Alaska is sort of our center of operation. It's about 70 to 80 miles south of the Arctic Circle. The Inupiaq Eskimos who inhabit this area, these are really the original Atkins diet people that in the traditional diet, there is no source of carbohydrates. So they rely entirely on marine mammals and fish for their diets. However, now, of course, with modernization and such, that there is access to additional foodstuffs. But I think it's worth pointing out, this is actually the store shelves in the one store that exists in Nome. And this is actually a real cornucopia compared to what exists in the small stores in the outer villages. But from the standpoint of trying to look at genes, the genetics of risk for these complex diseases, and trying to incorporate the diet contribution to that, we have sort of a natural experiment in that people living in this area don't have access to as wide a range of foodstuffs as most of the rest of us do. So basically whatever cooking oil is stocked is what everybody in the village has to choose from. Whatever canned goods that are stocked is the one thing everybody has to choose from. So it's not like there's a lot of choices that can be made. So the only real differences with individuals is how much they consume of the various components. So one of the things we've done in this study is we've done a fairly extensive profile of the fatty acids in both plasma and red blood cell fatty acids, and have done the linkage analyses of these now. And what I show you here is actually the strongest result thus far. And what's interesting, this is on chromosome 8, and that we get a very strong peak here, about 3.5 for one of the fatty acids in particular. But interestingly enough, there's a whole cluster of fatty acids in this particular region. So this, I think, gives additional support to the idea of a gene in this area contributing to what we're seeing. And when we look under, again, that linkage signal within the 1 log unit support interval, there are actually several very promising positional candidate genes. And so what we've done, of course, is go in and across that region, we've now typed 239 as SNPs, and have gone back and typed all of those in the individuals in the study. And so doing measured genotype analysis, this is basically association analysis done in the context of families. But what we find is that we have highly significant p-values for association with variation in a number of these genes in this region with our phenotypes of interest, some of which also include some of the measures of fat. So the beta-3-adrenergic receptor with fat in this case, BMI percent fat with apolipoprotein J, lipoprotein lipase with triglycerides and unsaturated fatty acids. Macrophage scavenger receptor 1 shows for a number of the poly and monounsaturated fatty acids in this region with a significant association. And then the tumor necrosis factor superfamily member 10 beta also with a fairly significant one as well. And I think one of the things, again, that makes this type of analysis so possible is the fact that we don't have lots of confounding of people eating lots of different diets. There really is no access to fast food establishments, anything else. So you're pretty much limited in your choices. One of the other things we've looked at in this sample is the genetics of delta-5 desaturase, and in both red blood cells and plasma, the blue shows the red blood cell signal, the pink shows the plasma signal, but they localize to the same region, and the red blood cell one, of course, has a very strong linkage signal there. Now, interestingly enough, in this region, there are 4 potential candidate genes that in some ways aren't too surprising, but are of interest. Fatty acid desaturase 1 and 2, as well as 7-dehydrocholesterol reductase and carnitine palmitoyl transferase. We've gone in and we've sequenced now, or we've genotyped SNPs in these 3 genes, 16 in the FADS1 and 68 in FADS2, 30 and 98 in the remaining 2. And again, doing a measured genotype analysis of these, what you can see is we get highly significant p-values for a couple of those different components. So p-values in the association of 10 to the minus 26 and the highest down to about 10 to the minus 5th. And since at this stage, this is not a genome-wide analysis any longer, but it's a positional candidate based on genome-wide scores, you no longer have to have that 10 to the minus 6th or 7th to be significant. So these well surpass even marginal significance and stand up to any type of Bonferroni's multiple testing, if indeed that were going to be an issue. just more of the same. As you can see here again, this is for the plasma measures, and again, very striking p-values, 10 to the minus 28th to the 23rd, 39th, and such. So currently with this, the desaturase work in this population, we're in the process of going in and doing deep resequencing across these 3 genes in the founders of the study itself to make sure we capture all of that variation, and then we'll go back and retype all of the SNPs we identify in the total sample of over 1,200 individuals. One of the other things we're doing, of course, is attempting to replicate these findings and working again, going back to the San Antonio Family Heart Study, where I started about 3 years ago now. We completed our transcriptome profiling in this population as well. And that's turned out to be a real gold mine, because what we've been able to do is look at the genetics of transcription levels across the entire transcriptome, and particularly looking for evidence of cis-regulation of particular genes. And I can tell you that currently what we've found in this case is that for FADS1 and FADS2, that they're both under very strong cis-regulation in the San Antonio Family Heart Study, and preliminary association analyses with FADS1 and FADS2 with a variety of the phenotypes we've looked at in the Gokadan study, looking at in the San Antonio Family Heart Study, are also showing significant evidence of association. So I think we've got very strong evidence here of these genes being involved in these regulations. So I want to finish by going back to sort of what was the grand parental study for all the projects that we have going, and that's our diet and genotype in primate atherosclerosis. And we've heard lots of talk about other animal models, but I don't think anybody's really talked very much about non-human primates. So my institution also is one of the 8 NIH-funded primate research centers, and I'll put a plug in terms of my role as group leader for chronic disease and genomics, that anybody who would be interested in perhaps utilizing non-human primates in their studies, we welcome certainly talking with you and seeing what we could do. But talk a little bit here about the baboon. Like I said, it's been used in the research for atherosclerosis, obesity, and diabetes now at our institution for well over 30 years. And the baboon, out of all the non-human primate species, I think is quite unique with regards to its utilization in metabolic disease research. Humans and baboons both evolved on the African savannas, and one of the things that is unique about our dietary adaptation in both cases is, unlike so many of the other primates, that both baboons and humans are incredibly omnivorous species. That is, we will eat anything, and baboons, believe me, will eat anything. But that is a very different dietary adaptation than what most other primates have. Most other primates have a fairly limited, fairly specialized diet. And so I think from that standpoint, it makes it a nice model. From a genetic standpoint, the thing that's unique about it is if you compare the entire genome, the total genome of the baboon and the total genome of the human, there's a conservation rate of about 95 to 96%. But if you move to just comparing the coding regions of the 2 genomes, that conservation jumps up to about 99%. So effectively at a protein level, the 2 species are almost virtually identical. And indeed, and when we do the assays in the lab for the baboon measures, we use the human assay kits with about 99+% success rate in that. So one of the signals I want to end up here with is, again, in our baboons. And one of the nice things, this is a large animal, which means it is possible to get repeated biopsies of the type we'd like to get in humans, but we're not typically capable of doing in a large genetic study. So I do have omental adipose tissue biopsies. One of the phenotypes we've looked at is both cell volume and cell size. And these are the results from a bivariate linkage analysis. So we're considering 2 traits simultaneously in the linkage analysis. In this case, PI1 levels, which is pro-inflammatory cytokine produced in adipose tissue, as well as fat cell size or fat cell volume in this case. And when you look at the two curves, the dashed ones here, these are the 2 traits independently. The dark line is the result from the combined linkage of the bivariate linkage analysis, and you can see in the combined state, again, we get a very significant linkage signal, well above 4. And so again, if we look at the 1 log unit support interval for this maximum linkage signal, there is indeed a very interesting, very promising positional candidate gene in that location, and that's this cannabinoid receptor 1 gene. So I think most people are familiar with the fact that cannabinoid receptor 1 is well recognized as a potential, having a potential role in the regulation of obesity, particularly through regulation of food intake. The drug rimonabant, which unfortunately was just withdrawn from use because of some of its other side effects, did show promises of being a very effective pharmaceutical treatment for obesity through limiting or affecting diet intake. But in our population of baboons, what we did, we went back, we completely resequenced the CNR1 gene in about 4 kb of the promoter region in 20 of the founding members of the population. We identified 12 SNPs in this way and took those 12 SNPs and then genotyped them in a cohort of 450 of our animals, and went back in and did, again, a measured genotype analysis. One of those 4 SNPs gives a highly significant association result, p-value, you see here, of 1 × 10⁻⁴. So again, very strong evidence of the cannabinoid receptor gene in these animals playing a role in regulation of obesity-related phenotypes. So I'd just like to, in conclusion, come up with a couple of points again. Again, this is sort of my dogma, but I feel very strongly that the quality of phenotyping becomes a very critical aspect for all genome-wide efforts. And I think that the better job we do of defining, classifying, and measuring our phenotypes, the better our chances are of finding meaningful genetic signals. I think there's also always important to point out there is utility in both case-controlled GWAS studies as well as family-based linkage studies. And that as we go forward, I think we're going to see much more of a merging and reliance on each of these techniques to sort of back up and to help sort out the results from each of the 2 different methods. The couple of, I think, interesting observations comes from one, the results we've seen with our POMC gene in the San Antonio Family Heart Study, where we actually have 4 relatively rare variants, but those 4 variants in various combinations can actually produce relatively common effects on the phenotypes of interest. I also think it's interesting and worth repeating that in the case of melanocortin-4, the example of the fact that some of these genes may have variants that cause relatively rare but massive effects, as in the case of some cases of childhood obesity, while other variants within that same gene can contribute to the more garden variety variation that we typically observe. And I also think that unique populations of humans, like our Eskimos, as well as non-human primates, offer ideal opportunities for genetic investigations where we can control or at least limit the variation that we see in the environment and allow us to look at some of the things that aren't typically able to do in a free-ranging and free-living population. So none of this work, of course, is done by one person in particular. I'm very lucky to work with a very good group of people. Particularly want to point out Dr. Saroja Vorigante, Dr. Paul Higgins, and Dr. Michael Profit, whose much of the work that I've presented today is a result of their efforts. And then also a large number of collaborators and colleagues, both at my foundation, or my institution, as well as a number of other institutions around the country, particularly Barbara Howard and Jason Newman, Sven Ebbison, my colleagues working in the Elaskimo project, particularly Sven. I fortunately don't have to spend much time up there during the colder months. Sven goes regularly. Unfortunately, I do have to go in January. Somehow I've offended somebody, I think. So I'll stop with that.